Так. Надія Вікторівна, можемо починати теоретично? Чи звук?
Так,
Починаємо чи ще?
YouTube підключили,
Угу.
запис ведемо, можемо починати. Так, всі на місці.
Можемо починати. Добре. Шановні колеги, dear members of the ad hoc specialized scientific council, dear guests. We meet today as an international team of the one-time ad hoc specialized scientific council, serving as a doctoral committee, and I, Alex Gumovsky, am acting as the chairman of the council. With the purpose to make our meeting accessible simultaneously for Ukrainian and English-speaking participants, I will switch from Ukrainian to English and back from time to time. First of all, I would like to introduce in both languages the members of our doctoral committee. I'm the chairman of the doctoral committee, Professor Alex Gumovsky. Я голова ради Гумовський Олексій Васильович, доктор біологічних наук, професор, член-кореспондент Національної академії наук України, завідувач відділу систематики ентомофагів та екологічних основ біометоду Інституту зоології імені Шмальгаузена Національної академії наук України. Members of the doctoral committee. First, reviewers. Dr. Yuri Kuzmin, Doctor of Biological Sciences, leading researcher at the Department of Parasitology, Schmalhausen Institute of Zoology, National Academy of Science of Ukraine. Yuri, please make us sign as you are here.
Thank you.
Yeah, Кузьмін Юрій Ігорович, доктор біологічних наук, провідний науковий співробітник відділу паразитології Інституту зоології імені Шмальгаузена Національної академії наук України. Оксана Шатковська, candidate of biological science, senior researcher at the Schmalhausen Institute of Zoology, National Academy of Sciences of Ukraine. Оксана, make us sign. Оксана Шатковська, кандидат біологічних наук, старший науковий співробітник Інституту зоології імені Шмальгаузена Національної академії наук України. And our dear opponents. Maria Freitas Neri. Hi, Mariana.
Hello.
PhD in Systematics and Ecology, Associate Professor at the Department of Genetics, Evolution, Microbiology, and Immunology of the Institute of Biology, State University of Campinas, Brazil. Am I right? Did I list everything correctly? Thank you, Mariana. Mariana Freitas Neri, співробітник лабораторії систематики екології, асистент на кафедрі генетики, еволюції, мікробіології та імунології Інституту біології Державного університету Кампінас, Бразилія. Rachel N. Rasico. Hi, Rachel.
Hello.
PhD in geology and geophysics, and head of the imaging laboratory department of the Messel Research and Mammalogy, Senckenberg Research Institute, and Natural History Museum, Senckenberg Gesellschaft für Naturforschung, Deutschland, from Frankfurt, right?
Yeah.
Okay. Тобто Рейчел Ан Расіко, доктор філософії в геології та геофізиці, завідувачка лабораторії зображувальних технік відділу або кафедри відділу Месельських досліджень в теріології Сенкенбергського інституту і природничого музею у Франкфурті. Thank you for your time and readiness to participate. Now I will declare some basic required statements in Ukraine, which correspond to our further procedure. Відповідно до порядку присудження ступеня доктора філософії та скасування рішення разової спеціалізованої вченої ради закладу вищої освіти наукової установи про присудження ступеня доктора філософії, затвердженого постановою Кабінету міністрів України номер 44 від 12 січня 2022 року із змінами, внесеними згідно з постановою Кабінету міністрів номер 341 від 21 березня 2022 року і номер 502 від 19 травня 2023 року та номер 507 від 3 травня 2024 року утворено разову спеціалізовану вчену раду Інституту зоології імені Івана Івановича Шмальгаузена НАН України з правом прийняття до розгляду та проведення разового захисту дисертації Телізhenko Валерії Сергіївни на підставі прилюдного захисту дисертації за темою Genetic and Morphological Proxies for Evolutionary Changes in the Ontogeny of Cetacean Skeleton, а саме генетичні і морфологічні індикатори еволюційних змін онтогенезу скелета китоподібних. На здобуття ступеня доктора філософії з галузі знань 09 біологія за спеціальністю 091 біологія. Науковий керівник, доктор біологічних наук, провідний науковий співробітник відділу еволюційної морфології Інституту зоології імені Шмальгаузена Національної академії наук України, професор Гольдін Павло Євгенович. Павло Євгенович. Публічний захист дисертації проводиться на засіданні разової ради за допомогою засобів відеозв'язку у режимі реального часу, а також відбувається онлайн-трансляція. Склад разової спеціалізованої вченої ради затверджено наказом Інституту зоології імені Шмальгаузена Національної академії наук України номер 18 від 26 червня 2025 року на підставі рішення вченої ради інституту, протокол номер 2 від 24 червня 2025 року. Науково-організаційний супровід засідання та підготовку документів разової спеціалізованої ради здійснює відповідальний секретар разової спеціалізованої ради Золотова Гайдамака Надія Вікторівна, кандидат біологічних наук, науковий співробітник відділу еволюційної морфології Інституту зоології імені Шмальгаузена Національної академії наук України. Надія Вікторівна.
Так, доброго дня.
Yeah, I must switch shortly to English back. I would like to introduce Nadia, who is in charge of our recording procedure and in general, the documentary flow work and other activities associated with this PhD defense, and in general, in charge of the procedure. So she is in the left upper corner. Back to Ukraine. Шановні колеги, у воєнний час пропоную узгодити дії на випадок повітряної тривоги. Залишайтеся на зв'язку зі мною, пане Надією. Засідання ми продовжуватимемо лише, якщо дисертант та всі члени ради перебувають у безпечних місцях. Інакше оголошуємо технічну перерву без переривання трансляції і продовжуємо після закінчення тривоги. What was my last sentence is that in the context of martial conditions, I propose that we agree on the actions in case of the air raid alert. Please stay in contact with me and Miss Nadia. The meeting will continue only if the candidates and all council members who are in Ukraine currently are in safe locations. Otherwise, the technical break will be announced without interrupting the live broadcast, and the session will resume after the alert ends. But we hope nothing will happen during this couple of hours. Back to Ukraine. На засіданні присутні дисертант, науковий керівник і всі 5 членів ради. Є кворум, і таким чином ми можемо почати. Чи нема заперечень щодо цього серед колег? Зауважень? Якщо немає, то тоді ми починаємо. So, the candidate, the scientific supervisor, and all 5 council members are present, and we have the quorum, so we can start. Any objections, colleagues? If no, then we proceed. Then I declare the meeting of our ad hoc one-time doctoral committee open. Засідання разової спеціалізованої вченої ради оголошую відкритим. So the main and the first question, well, not the main, but at least first, we have to agree on the language we are going to use. again in Ukraine, необхідно погодити мову, якою буде здійснюватися захист дисертації. Пані Валерія, які ваші побажання щодо мови захисту? Valeriia, what kind of language would you like to use?
Так, I would like to use English.
І українською для протоколу?
Я би хотіла використовувати англійську. Дякую.
Upon the agreement with an applicant, the defense will be conducted in English. За погодженням із здобувачем, захист буде відбуватися англійською мовою. So we will conduct our PhD defense in English. Nevertheless, I declare some formal data about the applicant and the documents submitted by her beforehand in Ukrainian. So now will be a relatively big part speaking in Ukrainian. Дозвольте довести до наукової спільноти дані про здобувача. Телізhenko Валерія Сергіївна народилася 1994 року в Луганську. В 2020 році закінчила Національний технічний університет України, Київський політехнічний інститут імені Ігоря Сікорського і отримала повну вищу освіту за спеціальністю біотехнології та біоінженерії. Диплом магістра з відзнакою. Аспірантуру проходила у відділі еволюційної морфології Інституту зоології імені Шмальгаузена Національної академії наук України за спеціальністю 091 біологія, навчання в якій успішно закінчила в 2025 році. Освітньо-наукову програму підготовки здобувачів 3-го освітньо-наукового рівня вищої освіти спеціальності 091 біологія виконала успішно. Тема дисертації Genetic and morphological proxies for evolutionary changes in the ontogeny of cetacean skeleton, генетичні і морфологічні індикатори еволюційних змін онтогенезу скелета китоподібних, затверджено вченою радою Інституту зоології Національної академії наук України 24 вересня 2024 року за протоколом номер 8. Основні результати дисертації, висновки та пропозиції знайшли відображення у 3 наукових статтях за темою дисертації у наукових виданнях, що індексуються в базах даних Scopus, перший квартиль та або Web of Science Core Collection, а також в неї є 6 тест-доповідей на науково-практичних конференціях, 5 з яких міжнародні. 13 травня 2025 року протокол номер 6 на розширеному засіданні відділу еволюційної морфології Інституту зоології імені Шмальгаузена НАН України дисертацію Телізhenko Валерії Сергіївни було рекомендовано до захисту. 24 червня 2025 року вченою радою Інституту зоології імені Шмальгаузена НАН України ухвалено рішення про утворення разової спеціалізованої вченої ради Інституту зоології з правом прийняття до розгляду та проведення разового захисту дисертації Телізhenko Валерії Сергіївні на здобуття ступеня доктора філософії з галузі знань 09 біологія за спеціальністю 091 біологія. 26 червня цього року наказом Інституту зоології за номером 18 створено разову спеціалізовану вчену раду з правом прийняття до розгляду та проведення разового захисту дисертації Телізhenko Валерії Сергіївні на здобуття ступеня доктора філософії з галузі знань 09 біологія за спеціальністю 091 біологія. Інформацію про утворення разової спеціалізованої вченої ради внесено до інформаційної системи на ЗЯВО 26 червня 2025 року під номером ID 9962. Рецензії від рецензентів, відгуки від опонентів надійшли вчасно та зареєстровані в науковій частині Інституту зоології 19 серпня 2025 року. Таким чином, подані дисертантом до разової спеціалізованої вченої ради документи надійшли в повному обсязі та відповідають встановленим вимогам порядку присудження ступеня доктора філософії. Dear colleagues, we are back to English, and we can conclude that the documents submitted by the applicant, and this was the list which I just announced, to the one-time specialized doctoral committee have been received in full and meet the established requirements of the procedure for awarding the degree of the Doctor of Philosophy. So, for our further procedure, I propose the following schedule. 20 minutes for the candidate's presentation and 5 minutes for the questions and answers. Do we agree on it? Great, thanks. So the agenda is approved anonymously, and to present the main points of the dissertation, the floor is given to the candidate, Valeriia Telizhenko. Please, Valeriia, you can start your presentation.
Thank you. Let me share my screen. And you see my presentation?
Yes, not yet in a full mode, I guess. Yeah, now in a full mode.
Yeah, everything is working. Okay,
Yes.
so I will start. Good morning or afternoon, committee members and colleagues. Today, I will present my dissertation research titled Genetic and Morphological Proxies for Evolutionary Changes in the Ontogeny of Cetacean Skeleton. Let's begin with the broader evolutionary context. In the process of transitioning to a fully aquatic lifestyle, cetaceans underwent numerous modifications of anatomy, physiology, and life history. The morphological transformations of cetaceans are characterized by significant temporal changes in postnatal ontogeny, including delayed sexual maturity, prolonged growth, and the retention of juvenile anatomical traits. Thus, the study of genetic and morphological indicators of changes in the rates of cetacean ontogenesis can greatly contribute to our understanding of the processes of evolution and development in general. The aim of this work was to explore the morphological and genetic signatures of evolutionary changes in the skeletal ontogeny of different species of cetaceans. Objectives. First, to characterize phylogenetic trends in cranial suture ossification and test its possible correlation with the presence of accessory skull bones in cetaceans and terrestrial artiodactyls. Second, to conduct quantitative analysis of the evolution and genetic basis of diversification of four-limb skeleton morphologies in cetaceans and other placental mammals. Third, to analyze selection processes in the regulatory genes involved in humerus development in relation to variation in humerus shape in cetaceans. And fourth, to assess expression of regulatory posterior hox genes in cetacean flippers, tail flukes, and the dorsal fin during the postnatal ontogeny. The object of the study is the nucleotide and amino acid sequences, elements of the skull roof, and postcranial four-limb skeleton of cetaceans. The subject of the study is the morphological changes in the cetacean skeleton and the underlying genetic markers. Scientific novelty. In this study, I quantitatively analyzed postnatal cranial suture ossification and the presence of accessory bones in cetaceans and compared them to terrestrial artiodactyls. I also examined the relationship between four-limb morphology and selection in limb-related regulatory genes across all cetacean taxa. My findings suggest that accelerated evolution in humerus-related genes may play a role in shaping humerus morphology. Finally, this study was the first to identify and analyze the expression of posterior hox genes in cetacean limb, fluke, and dorsal fin tissues, showing that their expression persists throughout the lifetime of these animals. This research was conducted at the Department of Evolutionary Morphology at the Smalkausen Institute of Zoology, National Academy of Sciences of Ukraine. It was part of the planned topic, which focuses on the formation of complex, highly integrated structures in evolution and ontogeny. The work was also supported by the National Research Foundation of Ukraine. Background. As the skeletons of extant cetaceans exhibit multiple evolutionary modifications. These include delayed or incomplete ossification of cranial sutures, four-limb modifications into flippers, and the loss of externally protruded hind limbs. The cranium shows extensive remodeling, including a process known as telescoping, where skull bones shift and overlap, resulting in significant asymmetry in toothed veils. The number of post-cervical vertebrae also varies among species, which has important implications for locomotion and body support. Cetaceans display both pedomorphosis and paramorphosis, heterochronic processes that respectively lead to the retention of juvenile traits or the exaggeration of adult features. Markers of pedomorphosis were observed in porpoises and Commerson's dolphin, such as delayed ankylosis of vertebral and flipper epiphyses, delayed fusion of sutures between cranial bones, and changes in skull shape. Paramorphosis can occur due to different scenarios, such as the extension of the growth period and earlier onset of growth, or an increase in the growth rate. For example, the prolonged shift in the offset time of limb outgrowth can lead to the hyperphalangy, as was shown in the embryological studies on pantropical spotted dolphin. For mysticetes, the heterochronic processes can also occur in opposite directions. Nevertheless, it is important to remember that both pedomorphosis and paramorphosis can occur at different levels within the same species or lineage. Specifically, limb development can be viewed as a test case to illustrate heterochronic processes in ontogeny. Many genetic factors regulate cetacean skeletal development. An important and well-studied subset of homeobox genes, the Hox family, plays a fundamental role in vertebrate embryo morphogenesis by providing cells with regional information along the main body axis. In addition to their role in body axis formation, Hox genes are among the key regulators of the development of limbs. During the early phase of limb development, the HOX-A and HOX-D genes are activated collinearly, with the 5' located genes expressed in the modistal and posterior regions of the limb bud. There are also many more genes regulating limb development, including genes involved in digit identity, joint formation, and for limb elongation. Research on cetacean genetics has focused on whole genome sequencing, selection analysis, and transcriptome studies. Comparative studies of cetacean genomes have provided insights into adaptation for large body size, cancer-resistant, and skeletal modifications. This research builds on previous funding and integrates genetic and morphological perspectives to uncover new evolutionary trends. Also, genetics of cetaceans remain a challenging area due to the relatively recent advancement of methods and the difficulty of obtaining high-quality material, which is especially true for RNA studies. And now I'm going to materials and methods. We compiled two morphological datasets, one for studying the evolution of cranial ossification and another for studying rib-related genes covering 62 and 88 species respectively. Details on specimen numbers are on the slide for reference. This slide covers gene and protein data. We analyzed sequences from 65 species for limb-related genes, including 25 target and 25 control genes. Additional data on HOX-D13 and genes linked to humerus development are also shown, and details are on the slide. 3 specimens were used in this study expression analysis: Hyperodon ampullatus, Phocena phocena, and Tursiops truncatus. Samples of ampullatus and Phocena were collected by the author, by me, with permission from and in collaboration with the Scottish Marine Animal Stranding Scheme, part of the University of Glasgow. These samples were collected in 2024 on the shores of the North Sea, Scotland, from stranded animals that had died within a few hours. Tissues from an equally fresh carcass of Tursiops were provided by the Mediterranean Marine Mammal Tissue Bank, University of Padua, Italy. Samples of connective tissue were taken from 3 locations in 3 species: tips of flippers, tips of dorsal fin, and tips of fluke. Skin samples were taken from fluke, except for Tursiops truncatus, for which only connective tissue was collected. Additionally, a carpal cartilage sample was taken from ampulla, Hyperodon. For the study of suture ossification and accessory bones, skulls were visually examined for the state of postnatal cranial ossification in 25 sutures, best characterizing the cetacean skull. Suture ossification was estimated, and the presence of full closure or obliteration at some part of the suture was used here as the indicator of full ossification. Presence of accessory bony elements and also clefts, which potentially could indicate a trace from a partially fused element or show an innovation, was visually assessed and recorded at each specimen. This slide summarizes our methods for analyzing phylogenetic signal and trait evolution. We reconstructed ancestral states for suture ossification and accessory bones, estimated phylogenetic signal using Blomberg's K and Pagel's lambda, and modeled evolutionary rate variation with EVO rates and BOM. We also tested trait correlations using phylogenetic logistic regression. Full details are on the slide. At the molecular level, selection refers to the differential fixation of genetic variants, especially nucleotide substitutions in DNA sequences. Selection is typically detected through comparative analysis of mutation patterns in protein coding genes and can be categorized into several main types: neutral, positive, and purifying. Our pipeline for selection analysis of these genes looks as follows. We first defined our hypothesis, that means selected our target lineages depending on morphology. where we define hyperstates like hyperfolin here and hypostates as decreased number of elements. Then we collected coding sequences, made alignments, reconstructed guide phylogenetic tree, and performed different selection tests using various software, such as PAML or Relax. For expression analysis, we extracted RNA from tissues preserved in RNA later by grinding them in a liquid nitrogen and using Trizol. To remove genomic DNA, samples were treated with DNase 1. RNA quality was checked with nanodrop, and only samples with good purity were used. cDNA was synthesized using random hexamers and oligo-DT primers, and gene expression was analyzed by qPCR, targeting Fox genes and hot-tip long non-coding RNA. Expression levels were normalized using the housekeeping reference gene. I'm going to the results, and the first part of the study is focused on accessory bones and ossification of the cetacean skull. In overall, cetaceans showed fewer fully ossified sutures than terrestrial artiodactyls. Our results indicate that cranial suture ossification shows a strong phylogenetic signal that suggests that closely related cetaceans have similar patterns of delayed ossification.
Valeriia, can we stop for a while because the sharing of the screen was interrupted?
Yes. Is it working?
Nadia, could you please change it? Надія Вікторівна, мені здається, що ми бачимо ваш екран.
Неначе все працює.
Everything's fine.
Працює? А чо-
Very
Так.
good.
А я теж бачу екран Zoom. Ваша
Я-
конференція запущена.
Да, я не
Так.
бачу. Я не бачу екран.
У мене Наталя Слуцька скрін,
І-
звідки-то з'явився.
Ну да, ми бачимо Укрнет, новини України. Я не знаю, чи це екран, але це точно.
Також, я також бачу Укрнет.
А ну,
Так.
зачекайте.
А,
Може, Наталя?
Все,
Зараз.
повернулося.
Все добре.
Мгм. Yeah, you can continue. Sorry.
We also found that accessory bones are commonly associated with decreased suture ossification, which indicates a potential evolutionary trade-off. The EVA rates model suggests the general slowing of evolution of rates of total suture ossification. The calculated rates are declining by approximately 6% every million years. There were also several lineages for which rate of evolution was higher than average, with marginally significant posterior probability. These lineages include ancestral branches of subfamilies Delphinina, Lysodelphinina, and Globicephalina. Also, analysis using BAM did not find any significant shifts in the rates of suture ossification evolution. It highlighted several species with relatively accelerated rates. These species, Orcella brevirostris and members of subfamily Lysodelphinina, were also identified by previous EVA rates method. The analysis showed significant associations between total rates of ossification and presence of accessory bones or clefts. Thus, species with less ossified skulls are more likely to have accessory bones or clefts than those with the more fully ossified sutures. To summarize, this is the first comprehensive quantitative assessment of cranial suture ossification and accessory bone formation in modern whales that explores evolutionary patterns such as phylogenetic signal and evolutionary rates of skull ossification. The results suggest that delayed cranial development, likely influenced by pedomorphic processes, accompanied by reemergence or retention of multiple ossification centers. Prolonged ontogeny, large brain, and functional adaptations for underwater feeding and echolocation could be possible correlates or drivers of changes in cetacean cranium development. Now we are going to evolution of genes regulating limb development. Cetacean forelimbs show a wide range of morphological variation, with some species displaying increased phalangeal counts, while others exhibit reductions. High diversity of phalangeal counts, including the development of additional phalanx in all digits, including digit 1 in Delphinopterus, digit 5 in Balenoptera and Platanista, and extreme hyperphalangy in Globicephala. Reduction in phalangeal counts in digits 1 and 5 in most other lineages. Heterodactyl limbs in all Neobalenidae, Aschrectidae, Balenopteridae clade, and a variable number of carpal bones. The topology of the phylogenetic tree was identical to the topologies found in recent comprehensive studies of cetaceans and all mammals. The tree included all the major cetacean clades, which is important for further analysis. All nodes, except one, were supported by Bayesian posterior probability of 1. We examined selection with PAML and 25 limb-regulating genes using the branch model. All of them was under purifying selection. However, 10 genes showed a statistically significant increase in omega for the cetacean clade in comparison to other mammals. 7 genes, EVX1, EVX2, and few of HOXD genes, were found to be under statistically significant relaxed selection, which means that evolutionary constraints on these genes have weakened and allowed more amino acid changes to accumulate without strong purifying selection. Then we ran branch tests for various scenarios using different morphological skeletal traits as guides to choose target lineages. For these models, the foreground lineages were selected based on ancestral reconstructions. For example, we have chosen all lineages for which number of phalanx in the second digit was more than ancestral tree as hyperstate or hyperphalangy, and then repeated the test for the lineages with number of phalanx less than 3. Thus, we indicated several different trends. First, no genes under positive selection were found. Taking into account that accelerated evolution or relaxation of purifying selection can also indicate adaptive changes, we reported all cases where omega values differed significantly between target and background branches. In general, hyperstates such as polyphalangy in digits from second to fourth were mostly associated with elevated omega values in EVX1, HOXD9, HOXD10, HOXD12, and GDF5 genes. However, for carpal bones, the trend was the opposite, and these genes were associated with decreased number of bones. The most consistent pattern in substitutions across aquatic mammals was the multiple insertions and deletions in the polyalanine tract in HOXD13 gene. We observed high variation and diversity in the length of the polyalanine tract at the 5' end of HOXD13 in all the cetaceans. The number of alanine repeats ranged from 10 to 18 in modern cetaceans. The highest count of 18 alanines among whales was observed in members of Cogidae, Phacinoides, and Delphinopterus laicus, while the lowest count with only 10 alanines was found in Susa chinensis. This contrasts with the highly conservative number of 15 in most other mammals, except several groups which also had an expanded polyalanine tract, such as Sirenians, Afrotherians, the naked mole rat, the great horseshoe bat, and the walrus. In the next study, we analyzed selection in 11 genes known to be associated with humerus development. In general, the main trend for selection in these genes was also the strong purifying selection. However, specific details of this trend varied across the target phylogenetic lineages for each gene. Using branch models on separate branches, we identified several lineages in which omega values significantly differed from the background. Among them, results for PTCH1 in Hyperodon ampullatus and Balena mysticetus were much higher than the background value. However, they did not reach cutoff level of 1, characteristic for neutral selection, meaning that selection acting on these branches was still purifying, but relaxed. The comparison of omega values between mysticeti and odontoceti clades showed significantly increased results in PBX3 for baleen whales, and additionally, odontoceti had higher values than mysticeti for the GLI3 gene. To summarize, the study offers novel insights in the evolution of the four-limb skeleton, including the analysis of correlations in evolution of anatomical traits and regulatory genes in cetaceans compared to other mammals, detailed ancestral tree reconstructions, and the identification of relaxed selection in key regulatory genes. The approach of using ancestral reconstructions to identify target lineages for selection analysis could be adapted to investigate gene phenotype correlations on a broader scale. And we are going to the final chapter. It's a study of expression. We identified and quantified the expression of posterior HOXD genes HOXA13 and HOTIP in the connective tissues of the flippers, dorsal fin and fluke, of 3 species, Hyperodon ampullatus, Phocena phocena, and Tursens truncatus. In Hyperodon, the highest expression levels were obtained for HOTIP, HOXD10, HOXD11, and HOXD13 in the fluke, dorsal fin, and carpal cartilage. HOX-A13 and HOX-D12 were either undetected or exhibited nearly zero expression. The expression levels of the second exon of HOTIP were lower than those of the first exon. While RNA activity in the tips of the flippers was minimal, it remained detectable for most genes, with the exception of HOX-D12. The patterns were partially similar in the dorsal fin samples of Phoceana. However, the expression levels of HOTIP, HOX-D10, HOX-D11, and HOX-D13 were higher than in the dorsal fin of Hyperodon. HOX-D11 increased sixfold, and the first exon of HOTIP fivefold, and HOX-D10 twofold. Additionally, HOX-D13 was highly expressed in the porpoise, but was almost undetectable in Hyperodon. In contrast, the other samples showed low expression levels ranging from minimal in the fluke connective tissue to almost absent. The lowest expression was observed in flippers. The expression in the Tursiops samples was different from that in the other analyzed specimens. Most genes showed very low levels across tissues, with HOX-D12 completely undetected. In contrast, both exons of HOTIP, especially the first, and HOX-D11 had extremely high relative levels in the flippers. HOX-D13 was also relatively increased, and HOX-A13 was detectable. This is the first study to report persistent postnatal expression of posterior HOX genes in cetacean flippers, dorsal fin, and tail flukes. The identification of unusual expression patterns point to the importance of further studies of gene expression in non-model animals, which could potentially shed light on processes relevant to human health and medicine. In conclusions, cetacean skull evolution is characterized by delay in future ossification during the postnatal ontogeny, which shows a strong phylogenetic signal among cetacean and terrestrial artiodactyls. Along with phylogenetic signal in situ ossification, several parallelisms are seen among cetaceans and terrestrial and artiodactyl lineages, such as deer. The evolutionary rates for situ ossification show tendency to slow down both for cetaceans and for terrestrial mammals. Artiodactyls, also several delphinidae lineages have accelerated rates. The delay in situ ossification is often coupled with the presence of accessory bony elements and cranium across several cetacean lineages. The appearance of accessory bones breaks the trend in reduction of the number of skull elements in vertebrates. Trends in reduction and addition of bony elements for the manus across evolutionary lineages in cetaceans and non-cetacean mammals correlate with trends of evolutionary changes in genes regulating lip morphogenesis. Genes regulating manus morphogenesis, including HOX genes EVX2 and GDF5, which are strongly constrained and are under purifying selection in most mammals, show numerous examples of relaxed selection in cetaceans. Cetaceans have multiple non-synonymous changes in limb-related regulatory genes, including insertions and deletions. Some of these cetacean-specific patterns, such as an expansion or in some cases a constriction of polyalanine tract in HOX-D13, are shared by several mammalian lineages, including other aquatic mammals and other taxa showing non-typical limb morphology. Trends of relaxed selection were observed for the genes involved in regulating humerus development. Additionally, two positively selected sites were identified in GLI3 coding sequence of Kogia breviceps. In addition, a stop codon was found in PAN-X3 gene sequence of Delphinopterus laicus. The relaxed selection in gene evolution, combined with insertions and deletions in regulatory genes and positive selection on specific sites, seems to be an important evolutionary pattern driving aquatic adaptations of mammalian limbs. And finally, the analysis of expression of posterior HOX genes in cetaceans showed the absence of HOX-D12 expression in adult animals. The expression levels in juvenile Hyperodon ampullatus and adult Phoceana were similar, with HOTIP, HOX-D10, HOX-D11, and HOX-D13 retaining expression in dorsal fin and fluke connective tissue. Finally, a senescent Aphelina dolphin had highly expressed HOTIP and HOX-D11 and moderately expressed HOX-A13 and HOX-D13 in the connective tissues of the flipper tips. These results highlight the unusual expression of these genes in adult cetaceans and suggest specific regulatory mechanisms. This research was made possible with support from my advisors, collaborators, and funding institutions. Thank you for your attention, and I welcome any questions.
Thank you. Thank you, Valeriia, for that interesting and nice presentation. And from now on, we switch to the questions section. So when everybody, and in particular the members of the committee, are welcome to ask questions. And I will use my privilege to ask question first. At least I don't forget what I wanted to ask indeed. So Valeriia, a couple of questions. First one, how the phylogenies you obtained correlate with established phylogenies of cetaceans? And in particular, if I'm not mistaken, I've seen that orca was in different positions in the cladograms demonstrated on 23 slide, I guess, and then on 25 and 32. So their position of orca changed. And whether the phylogenies, established phylogenies of cetaceans correspond to the position of the cetaceans you used in your analysis, and whether the patterns are the same. The
Thank you
first
for
question.
the
Yeah.
question. Yes, should
You
I
can
go
return to the
phylogeny?
slides to show them.
Yeah. The phylogeny actually totally correspond to the study of Michael McGowan, 2020. But yeah, the study was written in 2023, so probably I was not able to compare it with later publications. We used Bayesian methods, and posterior probability was all very good, except for one branch, for which it was also less than 1, but more than 0.95. And the topology for other mammals corresponds totally with the paper in 2023.
Yeah, can we check here position of orca here, for example, on 32, if slide 32. Like
That's 32.
here it is, and yeah, 32. Yeah, it is somewhere in the middle here. Yeah, Orcinus orca, and on the previous it was like close to the basal node, or as a sister group to
It's
other.
the same.
Same?
Yes. Between Lycopleurus acutus and
yeah, and probably the previous slide, the like yet before 25, maybe 23 or something. I'm not mistaken. Yeah, yet back. Yeah, like and yet back. Yet back probably.
This one?
23 or so, still back, not 25, but back. Not yet back. Yeah, maybe the first cladogram that you have.
In the study for
Yes.
skulls.
Yeah, like here, yeah. Orca was like a sister group to the rest of cetaceans.
Yes.
Right?
Yes, because for this study I did not reconstruct trees. I
okay.
took it
So
from
this was the cause of my question.
Okay, yeah, the study, it was the second one, and it was written later. So for this, the phylogeny was updated, and we took the newest sources instead of reconstructing it as the previous one.
So how can you generalize the differences in the position of orca in topologies? It's because of the methods applied, or there are some challenges?
I think it's because of more data used, like more genes or whole genomes.
Yeah, okay.
Instead of subset of genes.
Okay, it's just because of the limited fraction of the data analyzed, right?
Yes, I think.
Okay. Then the second question, you hypothesized the role of the ossification in the evolution of cetaceans. Did you compare your data and conclusions with a paleontological history of cetaceans? Is it possible to verify your implications based on the fossilized material? Other guests who would like to ask you questions. Thank you. So colleagues, please.
Can I go?
Yeah, sure, Rachel.
I think my question's kind of related. I was really excited, really great talk, by the way. I was really excited about your findings on the last chapter, also as you presented as well as in the actual presentation. But the really exciting thing is about finding that the Hox genes are expressed even in adults and even in a really old adult. And you mentioned in the paper too that it needs to be, sorry, the dissertation, that you need more data to confirm things, which I think is true. But you also have a really good like phylogenetic bracket, a good selection of taxa. And so like using that information, can you suggest like what you might find in other taxa because you have a delphinid, a physinid, and a ziphiid. Given that range of taxa, would you predict that this would also be the case in other groups, or do you think there's something special about dolphins and porpoises and ziphiids specifically? And then, so like basically using the phylogenetic bracket, what would you predict you'll find in other groups? And then I have a second part to the question, but I'll let you answer that one first.
Okay, yeah, thank you for your question. I think it's one of the complicated subjects because expression is a very like dynamic, and it can change not only with different species but in different life stages, as we can see here. So that's why we need more data. For example, these species, they were all of slightly different life stages. This Hyperodon, it was quite juvenile specimens, and Phocena was adult, and Tursops, it was senescent, like around 60 years old. So for next studies, we should probably compare species of the same age. And I suspect it could also be connected with, if we will look back to the study of limbs in some species, such as Hyperodon, the limb is more ossified and has more carpals compared to Phocena, which is more cartilage, has more cartilages. So maybe in analyzing other beaked whales, the results should be similar to Hyperodon. And when analyzing other Phocena species closer to this harbor porpoise, I suspect. It's better to have also some sequencing, transcriptomic sequencing data, I think, to
So
be fully sure.
would you be able to target those groups so you have kind of already an outline in your head of what you would do next for looking at them?
Yes, I would like first to check this Tursiops sample with different method, maybe some, I don't know, immunohistochemistry to detect if this protein, like genes are expressed at protein level, because the results were very weird. I
Yeah.
checked it, like I did several replicates, and the numbers are very high compared to reference gene. And then I would like to see, maybe make a transcriptome sequencing for the most available species. The main complication is that it's difficult to get fresh samples.
Oh yeah, for sure. I mean, given the sample you have though, I feel like it really means a lot. Like I don't know, I just because you have a diverse sample, I feel like it could still even say something evolutionarily, even though they're diverse in age and different groups that they're from. You can still say something evolutionarily about it, I think. And
Yes, after this work, after I already did this dissertation, I looked into expression from public data on NCBI. They have some data of sequencing different whales.
okay.
But they usually use different tissues, such as blood or liver. so I cannot directly compare these numbers.
Okay.
But these genes actually expressed also in bowhead whale, if I
Okay.
remember correctly, mostly in reproductive tissues, the same as in other species, as in human, but also in muscles. So
Oh.
yes, we probably need more data to say something like with high confidence.
Okay.
So Rachel, was there the two questions you wanted to ask?
Yeah, they were kind of intertwined together. So yeah. Yeah.
Okay, thank you very much. You are satisfied with answers of Valeriia?
Yeah, I have another general question. I don't
Yeah,
know.
for sure. For sure.
This is just like I'm pretty excited by this, but what are you excited to do next in your what are your kind of next plans and what are you most excited about doing next, I guess, in your research?
In exactly this research, yeah.
I mean, any of
Okay.
it doesn't have to be this. This is something that excites me, but it doesn't have to be. It's whatever you're excited about next.
Yeah, it's a bit of personal question because I honestly thinking about switching my field a bit.
Okay.
More into methodology, and like development of some mathematical methods to assess. But I can still use whales as my target subject for this.
Cool.
Okay. Any more questions, Rachel? No?
Yeah.
Okay, Mariana, your turn. Thank you.
Hey, thank you so much. Thank you, Valeriia, for the invitation, to Pavel as well. It was a pleasure.
Thank you for joining us.
Thank you. Valeriia, you write very well, and I think you have done an excellent work integrating morphological molecular approach. I do have a conceptual question. You've chosen to analyze protein coding sequences of highly pleiotropic developmental regulators, many of which are known to be among the most evolutionarily constrained genes in animal genomes because of their broad developmental roles. And you're seeking to link specific molecular signals to discrete phenotypic changes. And this creates a particular puzzling scenario when we consider specific cetacean appendage evolution, because the same genes regulate both forelimb and hind limb development. And yet cetaceans show dramatically different evolutionary trajectories for these structures. No, forelimbs were modified into specialized flippers, while hind limbs were essentially lost entirely. And how can chance in the same protein coding sequences that you chose to study could explain such opposite developmental outcomes?
Thank you.
How do you reconcile this apparent contradiction between the changing proteins that you can relate, you related to humerus or to hyperphalange, etc., with the loss of the hind limb, and would be proteins that would be regulating both?
Thank you for your question. It's a good question. And what I can say about this methodology is that we mostly used not very big subset of genes that are not exclusively, but associated with forelimbs. But we did not analyze genes which are known to be associated with the loss of hind limbs, such as we used TBX5, but we did not use TBX4 gene. And in previous studies, if I remember correctly, it was shown that there is a selection in TBX4 or some mutations which can be associated with the loss of hind limbs. So I'm thinking that this study is more like a stepping stone for this kind of research for me. And it makes sense to analyze a much more broader data sets of genes, like thousands of genes, and then make some multiple test corrections and to see its associations with different morphologies, such as I did in this case, where I used associations with different morphological states. I use them as target lineages. The same can be done for absence of hind limbs, for example. So that could be a good idea for further studies, I think, because there are different genes which are interconnected, and the change in one gene it can lead to changes in some morphological trait because of this network in which it interacts with different factors. It's quite a complicated study, and as far as I know, it's problem even in human studies when these genome-wide associations do not explain everything. So yeah, I think this approach could be scaled to more genes and to more interactions. And also we can take some regulatory elements into account to see if the rates of evolution in these regulatory elements are also accelerated or not. And then somehow summarize this, make some associations.
Well, okay. And what do you think alternative approach might better capture the molecular mechanism underlying this link specific versus link general adaptations? Because there are several genes there that are related to many other functions, and of course, if you choose a specific phenotypic character different from those that you test, and I know you applied some corrections because of the multiple testing, multiple hypothesis testing, but yet you still are using genes that, as you can see in your last chapter, are being used for many other features because they are regulators of other genes. So it's it will be very difficult to me to find specific genes that you can really link to specific adaptations in such character, because it involves, as you said, multiple genes in the sequence, in the coding sequence. What alternative approach could be used to capture specifically these molecular mechanisms that you could specifically link to specific phenotypic characteristics, other than focusing on the coding sequence of these transcriptional factors genes?
We can compare also regulatory and non-coding elements, changes maybe in promoter sequences. And we can use other methods as well, some molecular methods like comparing expression too.
And sure, you're right. But would you expect that the changes in expression or in the regulatory regions would be easier to connect to specific changes, thinking about the specific phenotype that you are looking at than the coding sequence genes? Or you think it would be, where do you expect the answer would be more linked to it? Expression, regulatory genes, coding sequences?
I expect that it could be in maybe some we can use some integrated approach to find some node genes in which changes is the most correlated I expect maybe in regulation more.
Why?
Yeah, that's a good question. There were some papers actually about limbs when researchers were doing some essays and looking on how these genes are expressed in zebrafish, or I'm not sure, and looking into how these regulatory sequences can change the development of limbs in laboratory settings. So yeah, maybe in regulation. But we need to just do more research, maybe in future, to be more sure.
Okay, thank you.
Mariana, you're satisfied with the answers of Valeriia?
Yes, I am.
Yeah, I think it was quite complicated scientific battle and interesting very much. Okay, other members of the scientific committee, Oksana, Yuri, who's next? Maybe Yuri?
Well, Oksana is raising her hand, so
Yeah. Okay. Then Oksana.
I will
First. Okay. Oksana, please, giving floor to you.
Is the relationship between the degree of ossification of the skull and the size of cetaceans. You report the high phylogenetic signal, but what about size of cetacean? Can slowing down ossification affect the skull size or cetacean as all?
Thank you. I think it could be also connected with size. I think these are all interrelated factors, because we have a theory that it may be somehow associated with feeding mode, for example, and we can see that in the big whales, in rorquals, which have this special feeding behavior. The skull is the less ossified of all, and all these whales are also one of the biggest mammals and the biggest mammals on the planet. And for some species such that exhibit more ossified skulls, such as the big whales, it can be also associated with some adaptations, such as diving adaptations and feeding.
Okay, Oksana, you're satisfied with the answer? You can ask more. Yeah.
Yes, I am satisfied with the answer, Valeriia. Thank you.
Any more questions from you, or you're okay?
Maybe.
Yeah, maybe one more.
One more. Can you explain in more detail how the appearance of accessory bones is related to echolocation?
Okay, appearance of bones related to echolocation. I'm not sure if it's related.
You reported that the present accessory bonds is related to echolocation and feeding in your dissertation.
I mean, all this ossification. Yeah, ossification together with the presence of accessory bonds, it can be connected with echolocation too. Because of changes in structure of skull.
Are there any studies which confirm some kind of role of accessory bonds in reflecting signals or whatever?
If I remember, yes, but I cannot answer right now because I honestly don't remember exactly about echolocation and accessory bonds.
We hope that your scientific advisor will help you because he will take his speech after all the questions. And yeah. Okay. And then other questions?
No, I don't have question.
Okay, thank you, Oksana. And then Yuri.
Well, yes, thank you. I thank Valeriia for brilliant presentation. And I have just one question, and it's almost on the same subject about the accessory bones. I'm just curious, is there any function of these structures, or they're just the signs of particular or incomplete ossification in crustaceans, in cetaceans? Do you know if there is any function of these bones?
I think it's more like a side effect of development, or maybe in some cases it makes skulls more like not separated and flexible.
You mean not so rigid?
Not so rigid, yes.
Yeah. Okay, thank you. I'm satisfied. Thank you very much.
Yeah. Is this the only one question, Yuri, or you would like to ask another one?
No. Just one.
Okay. So from now on, we have all members of the scientific committee with their questions and answers from Valeriia. And so from now on, we give floor to the scientific advisor of Valeriia, Dr. Pavel Goldin. Pavel, maybe you would like to tell something about your student.
Yes, thank you very much for giving me the floor, and overall thanks to all the members of the scientific committee for your inspiring and fruitful discussion of this paper. I like to say that Valeriia Telizhenko successfully completed her studies at the PhD school, and now, as you can see, she's presenting the fruits of her early research career. Valeriia showed herself as an exceptionally talented researcher. She's a highly motivated, multitasking, and hardworking student who conducted a fully independent research. Valeriia has been a true innovator who sets perfectly precise priorities and research questions to be addressed and finds the best optimal solutions. Valeriia Telizhenko combined morphological and data analysis in her research, which allowed her to review common patterns of evolution, the connection between anatomical transformations and changes in skeletal ontogeny, with transformations of morphogenesis regulation genes, using cetaceans as the main example, and to obtain the results of significant scientific novelty, which are important for the development of evolutionary theory. Here, I'd like to intervene a bit to the discussion and to the question which has been asked here. First of all, regarding the morphology and the possible role of delayed suture ossification in the cranium of cetaceans, it's an extremely good set of questions, and I suggest there might be several answers to these questions, because we see the parallel evolutionary processes occurring in mysticetes, in odontocetes, and even in some terrestrial artiodactyls such as deer. What is important to say that the family of Cervida, but not Regulida, as we expected in our zero hypothesis. And the possible explanations might be that for mysticetes and, for example, for sperm whales, for physeteroids, the delays in suture ossifications might be primarily associated with feeding, while in delphinids, for example, this delay might be primarily associated with the enlargement of the brain size, which can be especially well seen in orcas, for example. Orca, as well as the sperm whale, these are animals with the largest brain in all the animals, and it is spectacular to see that both species have also extremely delayed suture ossification of the skull. So the evolutionary direction, the constraints, and the drivers for this process might differ from between taxa and between species, but the result would be equifinal, and that's a good question to be further investigated. Also, another set of good questions about I would say, the opening Pandora's box, and what can we learn in future from the overall ideas of development and evolution of regulatory genes, how all this complicated system can work, and if we can find answer by further studying of non-coding elements such as regulatory elements and both short and long non-coding RNA sequences. Because in addition to long non-coding RNA elements, there are also microRNA elements and the whole set of regulatory elements. I cannot say for sure, and I believe that no one can say if we get an answer by further study of evolutionary patterns of these elements, but at least we will gain more knowledge by expanding our research on these elements. And as Valeriia said very well, we need a bit more development in our methodological bioinformatical apparatus for further development of such patterns of evolution, because you cannot just calculate a Bayesian evolutionary tree for example, for long non-coding elements. You should apply different methodologies which are maybe not so perfect as for today, and we should definitely need first better develop our methodological approach. And these are very, very good questions set up by this PhD project. And overall, I would say that Valeriia got the top important projects done, and she published her job as the articles in the Proceedings of the Royal Society B in the scientific reports, and also she was the co-author of the paper published in the Communications Biology. And she conducted approbation of her work at global scientific conferences, including several oral full talks. Valeriia has excellent, amazing quick learning skills and exceptional performance in research. She proved herself as a meticulous researcher, a geek, perfectionist who has been mastering interdisciplinary approaches, methods, and techniques of the cutting-edge bioinformatics, statistics, and mathematical biology, lab work, anatomy, and she has been equally perfect in computations, data sets, and work with biological material. During her studies, Valeriia achieved the highest professional level and gained enormous experience in international collaboration. I should express my deepest gratitude to this exceptional student who has brought to fruition the project that represents my life's work, and working with such a committed researcher has been one of the greatest privileges of my academic career. Thank you, Valeriia.
Thank you, Pavel. Thank you for that nice post introduction of your student. We got additional information and idea how the project was developed and what kind of plans and targets were stated by the scientific advisor and the student, and what was the background of the development of this PhD project. So now we are going on, and I must to declare that since the reviews of the reviewers and the responses of the official opponents have been published on the website of the Schmalhausen Institute of Zoology of the National Academy of Science of Ukraine in the manner and within the time frames established by the procedure for awarding the degree of Doctor of Philosophy, and they all are positive, there is no need to read them aloud. And in addition, the one-time specialized doctoral committee of the Schmalhausen Institute of Zoology of the National Academy of Science of Ukraine has not received any reviews or questions in writing or by email from other persons regarding the dissertation work of Valeriia Telizhenko. While the dissertation has received positive reviews overall, there are some comments and suggestions which have been raised by the reviewers and official opponents. And now I would like to give floor to Valeriia to answer the questions of the reviewer and opponents. The questions and comments will be highlighted on the screen, and Valeriia will answer, and the questions will be answered in this manner. So, Valeriia, I give you floor to answer the questions of the reviewer Yuri Kuzmin.
Okay, thank you. So the first comment is about the absence of authorities' names and years of descriptions in animal scientific names. And my response is, thank you for this observation. The authorities' names and years were not included with the first mention of species, and I acknowledge this omission, and I will take it into account in future publications. The main reason for this omission was that my papers do not contain taxonomic opinions, and I do not contest the current nomenclature. And including all the taxonomic authorities could substantially expand the list of references, which already had almost 400 reference items. The next comment is a suggestion to finish each section with a summary or conclusion. And I'm thankful for this suggestion and agree that concluding sections with a short summary can make the text clearer. Possibly for some pure introductory parts, such a summary would be would not be very meaningful, as these sections only provide background information not directly related to my research. But I agree that it is useful for the sections that are central to the dissertation. And as example, at the end of section 1.3, the following summary could be added. The summary is: research on heterochronic processes in cetaceans demonstrate the importance of both pedomorphosis and paramorphosis in shaping morphology, but findings remain inconsistent across lineages. While some studies suggest convergent pedomorphic traits in odontocets and paramorphic features in balenopterids, others dispute these interpretations, highlighting the need for integrative approaches combining morphology, ontogeny, and ecology. The lack of consensus shows that the role of heterochrony in cetacean evolution is still not fully resolved and requires further investigation. The comment for the chapter 3 is concerning Wormian bones and capitalization of the first letter in the term Wormian. And the term Wormian bones comes from Thomas Bartholin, who introduced it in honor of the anatomist Olaus Wormius. The word derives from a proper name, and capitalization is historically justified. For consistency, I tended to use Wormian bones with a capital W throughout the text, but somewhere the capitalization was lost during the text formatting. The second comment for chapter 3 is concerning abbreviations, which are sometimes their meaning is not explained in the first occurrence. And I agree that a separate list of abbreviations at the beginning should make a text easier to follow, and I will take this into account in future publications. The first comment for the chapter 4, and the only comment, is about formatting of several references. And I'm thankful that you're drawing attention to this, and this incorrect formatting is indeed could be the result of reference manager I used Zotero. I acknowledge that issue and will make sure to check and correct the references carefully in the future work. The comment for the chapter 5 is about inaccuracy, that I forget to change figure X to the number of this figure, and thank you for noticing that this inaccuracy. This reference to figure 33 was mistakenly left as figure X, and I acknowledge this error and will correct it. And finally, a comment for the conclusions, which are excessively extended, and I agree that these conclusions are longer with the others than the others. I made them more detailed intentionally, since these results are central to the dissertation and I wanted to emphasize their novelty. At the same time, I understand that the level of detail may look excessive in the conclusion section, and here I suggest a slightly shortened version while keeping the main findings intact. So the revised version of conclusion 3. Trends in the reduction and addition of manus elements across cetaceans and non-cetacean mammals correlate with evolutionary changes in regulatory genes for limb morphogenesis. Several of these genes, including Hox genes EVX2 and GDF5, show signs of relaxed selection in cetaceans with non-synonymous changes such as indels and polyalanine tract modifications. Some of these patterns are shared with other mammals, showing unusual limb morphology, and several positively selected sites within the cetacean clade were identified. And revised conclusion number 6. Posterior Hox genes show unusual expression in adult cetaceans. Hox D12 is absent, while hot tip Hox D10, Hox D11, and Hox D13 retain expression in dorsal fin and fluke connective tissue. Expression differences between juvenile and adult individuals, as well as the high expression of hot tip and Hox D11 in the senescent dolphin, suggests the presence of specific regulatory mechanisms that distinguish cetaceans from other mammals. The net discussion points highlighted in the review of the official reviewer Oksana Shatkowska. And the first comment is
Valeriia, wait a bit. We have to be reviewer and opponent separately making a pose. So I will have to ask Yuri, is he satisfied with your answers? Yuri, are you satisfied with the answers of Valeriia?
Yes, thank you very much, Valeriia. I am completely satisfied. Just one additional comment. The mention of authorities' names in the text would not expand your reference list because there is no need to include those names and year of publications of each taxon in the reference list. But all the rest is completely clear, and I'm satisfied. And thank you very much again.
Yeah, thank you.
Thank you, Yuri. Thank you, Valeriia. So now I give floor to Valeriia to start answering the questions and comments from the reviewer Oksana Shatkovska. Please, Valeriia, do it.
Okay, the first comment I will read it loud. The author focuses on postnatal development in the dissertation. However, many morphological traits are determined embryonically. Can the author findings be extrapolated to embryonic development? Thank you for this observation. The dissertation is focused on postnatal development, and I did not attempt to extrapolate the findings directly to embryonic stages. Embryogenesis is characterized by far more pronounced morphological transformations than those occurring after birth, as the body plan and major organ systems are established during this period. By contrast, postnatal changes are comparatively moderate and mostly concern growth and maturation of already formed structures. In my study of skull suture ossification, I chose postnatal material because it represents the final, most stable stage. During embryogenesis, sutures form and transform following very variable patterns across species and with individual variation, and the proper embryonic study would require dynamic sampling across developmental time points, which was outside the scope here. In a very rough sense, there may be correspondence. For example, skulls that are least ossified in adults and tend to be least ossified in embryos. However, embryonic development is much more complex and dynamic, and proper interpretation would require stage-specific data, including gene expression at defined developmental windows. In addition, other ossification processes than suture ossification can be seen in prenatal development. For this reason, the conclusion presented in this dissertation apply only to postnatal development, while embryonic development would require a separate dedicated investigation. The second comment, given that certain genes remain active into adulthood in cetaceans, unlike in other mammals, how might this extended expression influence limb morphology? My response, the morphological manifestation of extended expression of certain genes into adulthood in cetaceans can be considered only hypothetically, since direct evidence is lacking. In embryonic dolphins, hyperphalangy has been shown to result from a prolonged duration of limb outgrowth, which demonstrates how extended developmental timing can shape skeletal morphology. By analogy, the persistent expression that I detected in postnatal animals might be interpreted in several ways. First, as a continuation of growth-related processes. Second, as a residual signals from elevated expression during late embryogenesis. Or third, as an indication of some limited regenerative potential. These possibilities remain speculative and would require more experimental studies to clarify. However, importantly, the distal phalanx in hyperphalangeous cetaceans, such as delphinidans, ossify very late, consistent with the first scenario. In the very old dolphin, the highly elevated expression of some genes is more likely related to aging and maybe pathological processes, since similar expression patterns are known in certain human pathological conditions. Overall, the findings highlight further possibilities, but the interpretation remains theoretical and more research is needed to understand the full picture. The third comment is about the term reemergence that is used regarding ossification centers and cranial development. I want to clarify the usage, considering that evolution typically does not restore lost structures, but maybe replaces them with analogs or transformed derivatives. I agree that, strictly speaking, evolution usually does not restore structures once lost, but produces new untransformed derivatives. This is known as Dollo's law of irreversibility. It was proposed that unused genes, silent or pseudogenes, will lose their ability to be reactivated after approximately 8 million years and would certainly not last longer than 1625 million years. Nevertheless, some examples of violations of Dollo's law were proposed previously, such as in the case of the tooth revolution in vertebrates. For example, teeth in the lower jaw of the frog, or the reappearance of the molar in the Eurasian lynx. A detailed discussion of this topic would, however, be too extensive. In my text, I use the term reemergence in a descriptive developmental sense to indicate that some ossification centers became visible again at later stages of cranial development, after being reduced to or obscured earlier in ontogeny. I was not intended to suggest the evolutionary reappearance. And that's all. Thank you.
Thank you very much, Valeriia. And Oksana, are you satisfied?
Yes, I am. Thank you, Valeriia. I have only one comment. I think that embryogenesis and postnatal development are connected, and for example, number of phalanges, the presence of accessory bones is determined in embryogenesis, and postnatal development may be in postnatal development this structure may be different in size, but present this structure is determined in embryogenesis, and I think that these processes are more connected and maybe more detailed study in this topic is needed. Thank you.
Thank you, Oksana. Thank you for your nice comment. It will help Valeriia to strengthen future research efforts. And then we start answering questions and comments from the official opponents. And I give floor to Valeriia to answer the questions and comments from Dr. Mariana Freitas-Neri.
Oh. Yes, thank you. So the comment is concerning that the synthesis across datasets could be stronger, and certain methodological details require qualification for full reproducibility. Thank you for your comments. I understand the concern about synthesis across datasets. Since the dissertation is structured chapter by chapter, each part was presented with its own conclusions, and this sometimes made the overall picture less explicit. Still, I think the common topic of ontogenetic changes in cetaceans comes through, and in future publications, I will aim to draw these threads together more clearly. The same applies to methodological details. To keep the main text manageable, some steps were described briefly, while full datasets and scripts are available separately, and links to them are placed in the appendix to the thesis and supplementary data in the published research. There are 3 published research and links to them. I see that for a reader not directly involved in the analysis, more detail in the main text would make reproducibility clearer. This is a useful reminder for future work. Thank you. Yeah, this was the only comment.
Okay, quite interesting. And thank you, Mariana, for the comment. And are you satisfied with the answers from Valeriia?
Yeah, sure. Thank you.
Okay, thank you very much. And Valeriia, please follow on and answer the questions and comments from the opponent, Rachel and Rasiko.
The first comment for the chapter 1 is the suggestion to address the studies of Kum's paper about skull asymmetry that is associated with tooth well echolocation abilities. I'm thankful for this comment and agree that this could be added as following. Recent morphometric analysis confirms that cranial asymmetry in odontocetes is strongly tied to the evolution of echolocation. Coombs demonstrated that nasofacial asymmetry first became pronounced in early Oligocene odontocetes and increased in lineages occupying acoustically complex environments, supporting the interpretation of asymmetry as an adaptation to biosonar. Other recent research can be mentioned here. However, it is also true that many echolocating odontocetes show a decrease in asymmetry in their evolution, such as among the extant examples, Pontoporia, Orcinus, Phocanidae, and others, and the patterns of ontogeny of asymmetry highlighted by Coombs, Lancetti, and later are very promising for future research. The second comment for chapter 1 is concerning some organizational issues in the physiological adaptation section, where the paragraph begins discussing the cetacean stomach, but the entire paragraph centers more around cetacean sensory structures. Yes, and thank you for noticing this. It would indeed make sense to separate the mention of the stomach from the discussion of the sensory systems. The sentence on the stomach could be moved to a short note on digestive adaptation, and the rest of the paragraph would focus entirely on sensory modifications. The third comment for chapter 1 is that greater precision is needed in evolutionary terminology, and the example one sentence states Capera marginata is not a pedomorphic species, but species themselves cannot exhibit pedomorphism, only individual traits can be pedomorphic. I agree that the terminology should be used more precisely. In the case of Capera marginata, it would be more correct to say that the species shows or lacks some pedomorphic traits rather than calling the species itself pedomorphic. This will be taken into the account. The first comment for the chapter 2 is about methodology. Because it's about that usage of different phylogenetic signal metrics might be worth explicitly mentioning the Bloom. Okay, in the methods. Yes, thank you for the positive remark. It would indeed make sense to state explicitly in the method that Bloomberg K and Paget's lambda were used for continuous traits, in addition to D for binary characters, and I will keep this in mind for future work. The second comment for the chapter 2 about methods is about abbreviation ancestral AAS error. R that appears the first time in the phylogenetic tree reconstruction section without being defined. And yes, I totally agree. Thank you for pointing this out. It stands for ancestral state reconstruction, and it should be written in full at the first mention. I just forgot to add it. The first comment for chapter 3 about suture ossification, if I remember, that the chapter begins with methodological details rather than providing a brief motivation statement about why this research is important. And yes, I agree, the short motivation could be added at the beginning. As example, postnatal cranial suture development is a key aspect of skull growth that shapes morphology and function. Variation in suture closure reflects shifts in developmental timing, which provide important insight in the evolutionary history of cetacean cranial morphology. The second comment is about the more clear wording about early paragraphs discussing skull maturity, whether they describe the literal developmental ontogenetic stage of specimens examined or the results of the ossification analysis performed. And here I mean the results of the ossification analysis, as all the skulls are from the adult animals, if another ontogenetic stage is not mentioned in the text. It would be clear to say ossified rather than mature. The third comment for this chapter is about few terminology and clarity issues. And I agree that least ossified is not precise term here, since the study only scored sutures as either fully ossified or not. And what I meant is that certain sutures ossify less frequently across species, for example, those involving the nasal and frontal bones. The phrasing such as suture less often ossified may be clearer in this case. And the fourth comment is a suggestion to reorder suture-related topics or explicitly define in terminology to help readers follow. Thank you for this comment. I understand that discussion could be easier to follow if the order of suture topics was readjusted or some terms explained more clearly, and I will keep this in mind for future work. The fifth comment is about imprecise evolutionary terminology, and I'm thankful for this remark. I agree that in these places the wording may be not accurate. What I meant was that suction feeding involves a set of pedomorphic traits, and that some species show a concentration of such traits rather than being pedomorphic morphotypes. I will pay more attention to this terminology in the future. And the last comment for this chapter is a suggestion to reinforce my figure plot and spelling mistake in table 2. And thank you for noting these details. And I acknowledge the spelling mistake in the second table and the need for a clear representation of figure 11. As demonstrated on this slide where I highlighted species that are mentioned in the text. The first comment for the chapter 4 about limb morphology is also the suggestion to add a short description at the beginning. And as I agree, the introduction could be the next. The four limbs of cetaceans represent a striking case of morphological transformation with hyperphalangy, digit loss, and changes in carpal ossification, setting them apart from other mammals. These modifications raise questions about how developmental timing and gene evolution shape skeletal diversity. Here we study patterns of limb morphology together with the evolution of limb-related developmental genes to test how genetic changes may correspond to structural transformations. The second question is concerning the use of phalanx. The singular phalanx is phalanx. And the second part is about increase of phalangeal count in certain digits is paralleled by decrease in other digits, which is unclear. Thank you for this note. In the second case, I meant a decrease in number of phalanx of other digits, not a decrease of digits itself. The third comment for the chapter 4 is about adding a summary figure. And yes, thank you for this encouraging remark. I appreciate the suggestion, and while I was not entirely certain which figure was being referred to, I will certainly take this idea into account for future work and presentations. Because honestly, there was a figure about genes summarizing. I need to think more about adding other visuals. The fourth comment for chapter 4 is about wording again, which is unclear, and what I meant is better expressed as also EVX1 is clearly expressed in the limb, but its precise role in limb development remains unclear. The first comment for chapter 5 about humerus development is also suggestion to add some introductory sentence or paragraph, and I agree that the paragraph, this chapter could begin with the next. The humerus plays a central role in forelimb function, and its morphology shows considerable variation among cetaceans. Genes involved in humerus development therefore provide an important case for studying how evolutionary changes in regulation relate to skeletal diversity. This chapter analyzes patterns of selection in these genes across lineages with contrasting humorous morphology. The next comment is just about some technical correction, and thank you for noticing that transited should be transitioned. It's indeed the right suggestion. And the comment for the last chapter about expression, again, I agree that an introductory note could be added. For example, regulatory Hox genes and their non-coding regulator long non-coding RNA COTI play a central role in link patterning and tissue differentiation. Examining their expression in citations provide a way to assess whether postnatal expression patterns correspond to morphological changes of the forelimb and other appendages, such as flukes and dorsal fin. This chapter addresses variation in expression across tissues and age groups in odontosets. And another comment is also a technical note, is that common names are used once and not indicated which species these are, as species names are used throughout. And thank you for pointing this out. If common names are used, I agree it would be clearer to give the species name at the first mention, and that should be improved. And finally, comments for the conclusion, 2 comments, which suggest to make an explicit integration of how the different lines of evidence examined and make some synthesis of the common and connecting topics of the future work. Thank you for these comments. The conclusions in the dissertation are written chapter by chapter, but I agree that it would be useful to add a short synthesis showing how the different results fit together. I will keep this in mind for future publications. An integrative statement could be taken together. The results show that delayed cranial suture ossification, modifications in manus and humerus morphology, and unusual expression of limb-related regulatory genes all point to changes in developmental timing. These changes parallel signals of relaxed selection and positive selection in certain sites in regulatory genes, and thus both morphological and molecular evidence could point to heterochrony as a major factor in cetacean evolution. Yes, thank you for this comment.
Okay, thank you. Thank you, Valeriia, for the answers. And Rachel, are you satisfied with?
Yes, thank you.
Yes, thank you, Rachel. And now we are moving to the public discussion on the dissertation. During this part of our today meeting, each of members of the specialized council mandatory openly expresses his or her position on the awarding or refusal to award the degree of Doctor of Philosophy to the applicant, to Valeriia. Also, the experts being present at the meeting can take part in the discussion, to include means our guests, at their discretion. So, who wants to take part in the discussion first? We remember that members of the scientific council are mandatory to take part. Others, just voluntary. Who want to be the first? I can be the first if everybody thinks. Okay, so I will just say shortly because the dissertation was analyzed in details today, and I'm very impressed by Valeriia, by her contribution, by the mode of presentation, by the methods involved, and the extrapolations and the conclusions are made. I think it's a quite good and very impressive example of PhD thesis and the work of the PhD student who managed to do her best to reveal all the details and all the hidden mysteries of genetic background of morphology of cetaceans, and I believe that it will be great contribution to further studies on cetaceans, and it's very remarkable that Valeriia has managed to publish her achievements in a very nice first quartile reviews, and it's very impressive for any scientist and for the PhD student especially. I appreciate her work, and for sure I support awarding the degree of Doctor of Philosophy to her. Thank you. And who is the next? Probably Yuri, Oksana.
Oh, well, let me be the next, since I'm member of this committee. So I'm very impressed as well by the presentation, and it is very clear and very well structured. And the illustrations are beautiful in my opinion. And I would repeat the conclusion I made in the official review of this dissertation, that the dissertation is the author's original scientific investigation performed on sufficient material with important and interesting results and a high degree of scientific novelty. The results were included in 3 scientific papers published in international journals of high rank and presented at several international scientific conferences. And therefore, in my opinion, Valeriia possesses a high level of qualification as a researcher, and with no doubts can receive the scientific degree of the Doctor of Philosophy in the specialty 091 biology. This is my opinion, and thank you.
Thank you, Yuri. And Oksana, you are the next, please.
Thank you. I think we have had the results of very good research. The approach used in the dissertation is new for our department, and the combination of genetics and macromorphology is a very promising direction of research. Valeriia has obtained new data that significantly expand our understanding about cetaceans evolution. The works have many potential opportunities that we can see from the questions. The results of the study have been published in top-rated journals, and I support the opinion of colleagues and believe that Valeriia deserved to be awarded the degree of Doctor of Philosophy. Thank you.
Thank you, Oksana. Thank you very much. So our opponents, Rachel, Mariana, who is the first? Rachel, thank you.
Yeah. Yes, I agree with everybody. It was a really comprehensive and ambitious thesis with a lot of published papers. It was addressing really fundamental questions about how major evolutionary transitions are achieved through developmental programs and looking at everything from morphology to genes and different aspects of genetics. Were really it was a very comprehensive and very detailed and just pretty amazing piece of work. And yeah, and so I just want to say I didn't know that everything that I wrote was going to go through, but everything that I mentioned in my review was opportunities to refine rather than they didn't signify that there was anything particularly wrong with the work itself, because it was all really good and really interesting and innovative, I would say, as well. So overall, everything makes important contributions. I really hope to see some of the unpublished chapters out there at some point too. And but in general, yeah, I think the work merits the doctoral degree and recommend its acceptance, like my colleagues here.
Thank you, Rachel, and we will be glad to hear the opinion of Mariana.
Thank you. Well, I think this is really a strong and original dissertation that is advancing our understanding of cetacean evolution, integrating morphology, molecular evolution, gene expression. It addresses a fundamental question using complementary lines of evidence. The work and the answers that Valeriia presented today demonstrates methodological rigor, careful interpretation. Well, in my view, this dissertation meets and sometimes exceeds the standards for our PhD degree, so I support awarding the degree. Thank you.
Thank you, Mariana. And I just remind that now we had the public discussion. It's not yet voting procedure. It will be the next one. And before that, I will have to declare some formal statements for our procedure in Ukrainian. But beforehand, I would like to ask Valeriia, do you want to make a closing statement after today's presentations and questions and answers?
Yep, thank you. I would like to sincerely thank my supervisors, and everyone who contributed to the creation of this work. I hope that the findings I have shared today will contribute to the broader field and open pathways for further studies. I also recognize that research is always a work in progress, and I look forward to refining those ideas and continuing the discussion beyond the defense. Once again, thank you for your time, your engagement, and your thoughtful considerations of my work. Thank you.
Thank you, Valeriia. And now we go forth, and now we're approaching the voting procedure. The Council makes its decision by open voting according to our rules. I would like to remind that according to the minutes of the meeting of the one-time ad hoc specialized scientific council, meaning doctoral committee of the Schmalhausen Institute of Zoology of the National Academy of Science of Ukraine, which includes Dr. Alex Gumovsky, chairman, me, Dr. Yuri Kuzmin, the reviewer, Dr. Oksana Shotkovska, the reviewer, Dr. Mariana Freitas-Neri, opponent, and Dr. Rachel N. Rasico, the opponent, and all 5 members of the ad hoc specialized academic council were present at the meeting, including 2 doctors of science, 1 candidate of science, and 2 doctors of philosophy in the profile of the considered dissertation. So let's proceed to the voting. Colleagues, I would like to ask each of you to answer the question: do you agree to award Valeriia Telizhenko the degree of Doctor of Philosophy in the field of knowledge 09 biology in the specialty of 091 biology? It's just Ukrainian formal codification of these specialties. So voting takes place by raising the hand of each of the member of the doctoral committee, and also by the oral statement of this opinion, just that we are absolutely clear to the other committees which will be observing our activity and the validity of this PhD defense. So I ask step by step. So, Dr. Yuri Kuzmin, do you vote in favor of the awarding the degree to Valeriia, or against? Please make the statement clear.
Yes, I do. I vote in favor of awarding this degree to Valeriia.
Thank you very much. So, Dr. Oksana Shatkovska, you are voting in favor or against? Please make it clear.
Yes, I do. I think that
Hand. Make it by hand. Yeah.
that Valeriia deserves to be awarded the degree of Doctor of Philosophy.
Thank you very much, Oksana. So, Rachel Rasiko, please make your statement.
Yes, I agree that she should get the PhD.
Okay, thank you very much. And Mariana Freitas Neri, please make your opinion clearly, openly.
Yes, I vote in favor of Valeriia.
Thank you very much. And just not to forget myself, yes, I vote in favor of awarding the degree of the Doctor of Philosophy in the specialty of biology to Valeriia Telizhenko. Thank you very much. And yeah, we can preliminarily congratulate Valeriia, but we still have some formal procedures to follow. So, dear colleagues, so I can announce the unanimous decision of the one-time ad hoc specialized scientific council doctoral committee to award Valeriia Telizhenko the degree of Doctor of Philosophy in the field of knowledge of 09 biology in the specialty 091 biology, and we can congratulate Valeriia. Valeriia, congratulations.
Thank you.
Thank you for your presentation and your study. And now we have some formalities to finish, in particular to vote for the next for the next step, the text of the final decision of our meeting. I hope you all received the draft of possible decision of our doctoral committee on awarding the degree of Doctor of Philosophy in the field of knowledge 09 biology in the specialty 091 biology to Valeriia Telizhenko. Has everyone get acquainted with the draft? Have you seen the preliminary draft we have? Any suggestions or comments so we can keep it as it is?
I have
Yeah,
one comment.
Oksana, please.
В українській версії там у пункті 3 там щось написано падоморфні чи щось таке, там, мабуть, якась опечатка. Подруківка, потрібно його на педоморфні.
Yeah, there is a question about Ukrainian version of this decision. We deliver the question to Mrs. Nadia Zolotova Haidamaka. I hope you will be able to improve this small typo in our Ukrainian form. And I will currently I will read the text in Ukrainian, whereas the English version will be shown on the screen, so you will be able to familiarize yourself again with the text. Hope
Can I make a
Nadia.
small comment too, really quick?
Yes.
It has there's something about my date of graduation, I think, which is it's incorrect, but it's also not listed for other people, I think, so you could just leave it out. I don't know. It's a really small thing, but
yeah. Okay. And yeah, it's really nice. And okay, so now you have the text in English, and I will proceed with Ukrainian. And yeah, if there are any typos in English as well, just draw my attention to. And so in Ukrainian. Спеціалізована вчена рада Інституту зоології імені Івана Івановича Шмальгаузена Національної академії наук України ухвалила. Перше. Дисертація Теліженко Валерії Сергіївни на тему Genetic and Morphological Proxies for Evolutionary Changes in the Ontogeny of Cetacean Skeleton, which was submitted, що була подана на здобуття ступеня доктора філософії з галузі знань 09 біологія за спеціальністю 091 біологія, є завершеним самостійним науковим дослідженням і відповідає вимогам порядку підготовки здобувачів вищої освіти ступеня доктора філософії та доктора наук у закладах вищої освіти, наукових установах. затвердженого постановою Кабінету міністрів України від 23 березня 2016 року номер 261 та порядку присудження ступеня доктора філософії та скасування рішення разової спеціалізованої вченої ради закладу вищої освіти наукової установи про присудження ступеня доктора філософії, затвердженого постановою Кабінету міністрів України від 12 січня 2022 року номер 44 зі змінами, внесеними згідно постановою Кабінету міністрів номер 341 від 21 березня 2022 року та номер 502 від 19 травня 2023 року. Друге, на підставі результатів відкритого голосування разова спеціалізована вчена рада Інституту зоології імені Шмальгаузена Національної академії наук України присуджує Телізhenko Валерії Сергіївні ступінь доктора філософії з галузі знань 09 біологія за спеціальністю 091 біологія. В науковій частині інституту підготувати наказ про видачу Телізhenko Валерії Сергіївні диплома доктора філософії та додатка до нього європейського зразка, а відеозапис трансляції захисту дисертації буде доданий. So we are back to English, to our final stage. So dear colleagues, I have eventually a proposal to accept this decision of our doctoral committee as a basis. Who is in favor of this proposal? Please vote. Voting is done by raising your hands. I'm for. Everybody. Okay. Who are against? Nobody. Who has abstained? Nobody. Okay. Thank you very much. The draft decision is accepted unanimously. And I can again congratulate Valeriia on receiving her PhD. And I would like also to thank her supervisor, Dr. Golden, for his hard work as a scientific advisor, and thank to all members of our doctoral committee for their time, efforts, and great contribution to this PhD defense. Thank you, everybody. And I'm also grateful to all attendees and our guests and administration of the Institute of Zoology of Schmalhausen Institute of National Academy of Science for supporting our students and our research during these difficult war times. And all viewers for watching us, for their interest in our meeting and for their attention. Now I declare the meeting of the one-time specialized council closed. Засідання разової спеціалізованої ради оголошую закритим. Всім дякую. Thank you very much.
Thank you very